Okay, welcome back. This is Issues in Biotechnology, the way we work with life. It's the second part of the course on applications of biotechnology. And in the last lecture, we started medical biotechnology with a history of medical research, prosthesis development, cell culture, and so on, leading into stem cells and a discussion about how they're made and their sites of origin and possible therapies. And this dovetailed into techniques for animal cloning and the possibilities for human cloning and genetic engineering. In the second part, we looked at gene therapy, tissue engineering, and nanotechnologies that might apply to the future. In this series, we're going to examine pandemic flu, give a history and a context for what it is and how it occurs, and in the second part, look at bioweapons. The reason for putting these two together, I think, will become obvious when we look at dual-use knowledge. So we'll call this section then Biotechnology Applications in Tough Times, Pandemic Flu and Bioweapons. A pandemic is a worldwide outbreak of influenza. The picture shown here is actually depicting the plague as it affected Northern Europe in the Middle Ages, but it's appropriate for an image discussing pandemic. Recently, Hollywood put out a movie called Contagion, which I think is an interesting depiction about how a pandemic will play out. Will the specifics be as they are in this movie? Let's have a look at some of this. And we can describe some of these scenes. If you haven't seen this movie, it might be a useful backdrop. So this movie starts off with the woman talking to a friend of hers at a cough, a mere cough. She appears to be sweating a little bit. Let's fast forward this just a bit. She's hanging up the phone, and we see some other sick people, possibly in Southeast Asia. London, another sick person on mass transit. Fast forward a little bit further. A company report. A sick person on a train. A member of the CDC talking about a possible illness coming about. San Francisco. We cut back to the woman we saw in the beginning coughing. She eventually succumbs in a seizure, and this begins the story of Contagion. When we move further, her grieving husband, and then we see a meeting at the World Health Organization, and we meet one of the scientists who will become a protagonist in the story. We cut ahead to a meeting at the Minnesota State Health Facility, Department of Health. And they're discussing how to break this in the news. The scientist describes how a pandemic outbreak might occur. and says that this could be spread through fomites. And one of the press people asks her, "What do you mean, fomites?" And she says, people touch their face 2,000 or 3,000 times a day. They touch other things, water fountains, doorknobs, phones, fomites. She also describes how quickly a flu might travel. Again, the scientist at the World Health Organization. The reason why I'm describing this, in here, in this part, you see a biosafety laboratory level 4. And you see people walking around in what looks like spacesuits. Look at this piece of equipment here. It's a centrifuge. This depiction of this laboratory is not out of Hollywood. This is actually what a biosafety laboratory level 4 looks like. And the equipment is very accurate. In fact, let me just pause this a second, because the depictions in this film are all very accurate. They had well-known virologists work on the screenplay with them. And I'm myself, very persnickety about how Hollywood depicts science in general. And they did an excellent job in this film. Right down to the pipetters that they use in the lab and the descriptions that they use to describe how the genetics of this virus mutate. Let's go back to the movie just a second. So, the protagonist that I referred to earlier as a scientist is just describing the genetics in a scene here at the CDC. And I don't want to belabor the discussion right here for now. But how she describes the genetics that had to have occurred for this virus to jump from an avian source or some other animal host into human transmission is exactly what happens in the development of pandemic flu. Let's go back to our lecture. Is this science fiction? Well, it hasn't happened yet, but not in the way that Hollywood usually depicts science fiction at all. I have referred to this film as more of a documentary on something that just hasn't happened yet. How accurate is it? It's very accurate. It's accurate in its depiction of not only the science that occurs, but how a pandemic may play out in the United States and globally. And people ask me after they've seen this movie, this couldn't happen, really, right? I'm like, well, no, this actually really could happen. It's happened before, and it may not actually be a question of if, but really when, and a question of how prepared will we be. How worried should you be? Well, I think we should take this into account as something that could occur. So a pandemic is a worldwide outbreak of influenza, and an outbreak of a disease that occurs when a new influenza A virus appears or emerges in the human population. And it causes serious illness and widespread death, and easily spreads from one person to another. So that's basically what a pandemic is. And pandemics are different from the seasonal outbreaks of flu that we experience every year. Seasonal outbreaks are caused by subtypes of influenza virus that already circulate among people, whereas pandemic outbreaks are caused by new subtypes, by subtypes that have never circulated among people before, or by subtypes that have not circulated among people for a very long time. So we can look to history and see that past influenza pandemics have led to very high levels of illness, death, social disruption, and economic loss. There are many different subtypes of influenza or flu viruses. The species is influenza A virus of the influenza A virus A genus, or the Orthomyxoviridae family. All of the influenza virus subtypes are RNA viruses. They have a segmented genome of 8 negative-sense single strands of RNA, abbreviated as shown in this slide. There are 16 known HA subtypes and 9 known NA subtypes of influenza A viruses. The subtypes differ based upon proteins that are on the surface of the virus. The hemagglutinin, or HA protein, and the neuraminidase, or NA protein, are what constitute the H and N designations for the subtypes of influenza A viruses. Some have also been shown to differ in their polymerase genes. They have a low number of genes and are highly mutable. So the different sites of infection in a human more or less characterize their ability to spread and their lethality, as shown in this slide. The left shows an example of an H1N1 that is easily spread but rarely fatal, spreading through the lungs and upper nasal passage, whereas the H5N1 example shown to the right spreads slowly but often fatal. The rate of spread is actually dependent on the subtype and other conditions. So the genetic structure and related subtypes will influence the rate of spread of a pandemic and its danger to humans. The N in the H5N1, as I said, stands for neuraminidase, and the protein is depicted in this slide as a ribbon diagram. We now have the ability to rapidly genotype these viruses and to show the protein structure of both the H and the N components. Hopefully this gives us significant tools in the box in order to rapidly prepare when the next pandemic occurs. So the RNA strands that specify the structure of these proteins are most medically relevant as targets for antiviral drugs and for antibodies that may be developed in order to counteract a new emerging pandemic. And you can remember from our previous discussion in the pharmaceutical biotechnology section on vaccine development. You might want to go back at this time and review those slides in terms of the time it takes to develop a vaccine from start to finish, and its relevancy even to seasonal outbreaks. I gave a timeline about how surveillance is done, typically in the fall, and emergence depicted for the possible next seasonal outbreak by January, and then we would eventually, in the northern hemisphere, see vaccines on the shelf probably by late August or early fall. The H and N designations are also used as the basis then for naming the different subtypes of emerging viruses. The pandemic virus emerges as a result of a process called antigenic shift, which causes an abrupt or sudden major change in the influenza virus. So these proteins that are on the surface rapidly undergo mutation in their host, and this can influence their ability to infect both their avian host or other mammal hosts, including humans, and their ability to cross over from their host into humans. So changes then in these proteins or other genes result in new influenza A virus subtypes. So as the evolution of these viruses continues to occur, constant vigilance and surveillance is required in order to be prepared. Now, this certainly wasn't true earlier on in history, and we'll describe what the consequences of getting hit by something like this might be. Think of it almost like the 1938 hurricane that ravaged the Northeast and affected even here in Rhode Island, largely came ashore unannounced, whereas now we have long-range predictability, not that that can stop a hurricane or not that surveillance can stop a pandemic, but it helps to be a little bit better prepared. So the first step towards a pandemic, however, is that a new virus subtype must also have the capacity to spread easily from person to person. Surveillance usually involves noticing a new viral subtype in its host and its ability to be transmitted within its host. For example, if we notice a large number of birds dying, whether that's chickens or migratory birds, certainly this raises a red flag. Once a new pandemic influenza virus emerges and spreads, it usually becomes established among people and it moves around or circulates actually for many years as seasonal epidemics of influenza. So who actually is out there monitoring influenza viruses? Well, the US CDC, the Centers for Disease Control and Prevention, and the WHO have large surveillance programs to monitor and detect influenza activity around the world, including the emergence of pandemic strains of influenza virus. Let's take this into a historical perspective, that the movie Contagion did not come out of nowhere. During the 20th century, the emergence of several new influenza A virus subtypes have caused 3 major pandemics, all which spread around the world within a year of being first detected. Perhaps most notably and catastrophically was the emergence of the so-called Spanish flu in the years 1918 to 1919. That was a influenza A H1N1 virus. And then in 1957, 1958, the Asian flu, an H2N2 virus. And in 1968-69, the Hong Kong flu, which was an H3N2. And so you can see by the cycling of these that perhaps our situation is overdue now for a pandemic. Not that they come on regular cycles, but they come. So the Spanish flu caused the highest number of known influenza deaths since this has been historically monitored. However, the actual virus subtype was not detected during that pandemic. More than 675,000 people died in the United States in 1 year. at a time when the population was much less than what it is today. And up to 50 million, maybe more than that, died worldwide. So this thing went around the world like a wildfire. Many people died in the first few days after infection, and others died of secondary complications like pneumonia. But I think it's worth commenting here that we're all familiar with the effects of a seasonal flu outbreak. How you wake up maybe in the morning and you're feeling a little funny. You think that maybe you're getting sick. Maybe you go to work anyway, you think you might be able to fight it off. And then maybe that night you don't sleep well, you have a fever, and you struggle with this maybe for 10 days or so. And I would caution anybody that if you have those symptoms, stay home. Because likely you will only get into a public situation and infect other people. But also, remember our conversation on vaccines, that you might think that it's okay for you not to have a vaccine, but this is a herd mentality, and I did talk in a previous lecture about the cheater bird. Don't be a cheater bird. But in the case of the Spanish flu, if you had that funny feeling in the morning, and then you went home and started to feel sick, you might not wake up in the morning. This was very rapid, or if you woke up that morning, it might have only taken a few days. If you made it through that phase of things, then secondary infections and going to so-called tent hospitals further encourage the communication of secondary infections. So nearly half of those that died of the Spanish flu were young, healthy adults. So H1N viruses still circulate today after being introduced again into the human population as late as 1977. The Asian flu, an H2N2 virus, caused about 70,000 deaths in the United States. And it was first identified in China late February of 1957 and spread to the United States by June of 1957 and then had its catastrophic events during that year. The Hong Kong flu is an H3N2, and it caused about 34,000 deaths in the United States. It was first detected in Hong Kong early 1968 and spread to the United States later that year. This virus is still in circulation today. Another important clue to current events. Both the 1957 and the 1968 pandemics were caused by viruses containing a combination of genes from a human influenza virus and an avian influenza virus. The 1918 pandemic appears to have had an avian origin also. So, characteristic to the movie, Contagion, let's talk about what the stages of a pandemic actually are. How is it characterized? The WHO has developed a global influenza preparedness plan, which defines these stages of pandemic and outlines what the role of the WHO would be in recommendations for national measures before and during a pandemic. So during phase 1, no new influenza virus subtypes have been detected in humans. Surveillance is being directed primarily at animals, and that human infection may be present in animals. If present in animals, the risk of human infection or disease is considered to be low during this phase 1 of a pandemic. In phase 2, no new influenza virus subtypes have been detected in humans. However, a circulating animal influenza subtype poses a substantial risk to human disease. This is called the pandemic alert period. So if we notice a large number of pigs dying, or waterfowl, or chickens, this will raise the alert for a pandemic period. Phase 3 is when human infections with a new subtype have occurred, but there's no human-to-human spread yet, or at most very rare instances of spread to a close contact. Usually these types of interactions are first noted amongst agricultural workers, people handling birds, or perhaps first responders. Stage 4 is when small clusters occur with limited human-to-human transmission, but spread is highly localized, suggesting that the virus is not yet well adapted to humans. If you see the movie Contagion, there is a part when our scientist protagonist is describing the mutability of the genome in the virus that they're interacting with, and they see that now different subtypes have emerged, which is influencing the rate of spread in the pandemic that's depicted in the film. Phase 5, larger clusters of human infection occur, but human-to-human spread is still localized, foci, and an R0 is described. That is the ratio of people infected to the disease, suggesting that the virus is becoming increasingly better adapted to humans, but might not yet be fully transmissible. There's a substantial pandemic risk that's described during phase 5. This is actually what would be described as the beginning of the pandemic period. Phase 6, pandemic. Increased and sustained transmission in the general population, and the world is on fire. So a few notes here, that the distinctions between phases 1 and 2 is based on the risk of human infection resulting from circulating strains in animals. So we pay very close attention now to viral subtypes that are circulating amongst animals, and factors that include pathogenicity in animals are one point of research that is conducted by virologists that are involved in this type of work, and I'll get back to this later on in the lecture. The distinction among phases 3 and 4 is based on an assessment of the risk of pandemic, where various factors and their relative importance, according to current scientific knowledge, is considered. Mainly, what is the transmissible factor between model organisms for humans? That would be ferrets and perhaps primates, and I'll get back to that in a little while too. But also, again, monitoring animal transmission as it occurs in the public, between people who handle animals, and the presence of genes that might influence transmission from animal to human. Vaccines to protect against pandemic influenza viruses can be developed. And a vaccine probably, though, would not be available in the early stages of a pandemic. Why? Go back and review my timeline of how long it takes to generate a new vaccine. And again, Contagion does an excellent job of depicting how a new vaccine is made. I keep referring to our scientist protagonist in the movie, who endlessly, tirelessly is pursuing the development of a new vaccine. Meanwhile, the world's on fire. Her own father comes down with the disease. Finally, she thinks she has the vaccine. Her father's in the hospital. I don't want to be a spoiler here, but she injects herself with her own vaccine and goes to visit her father. I love it when scientists are finally heroes in Hollywood. So eventually, vaccine development will come down to manufacture, and then it'll come down to distribution. Manufacturers would have to use a selected strain to develop the vaccine. This is also true, you know, for seasonal virus. They choose the selected vaccine well before the emergent seasonal virus emerges. So once a potential pandemic strain of influenza virus is identified, it will take several months for a vaccine to be developed, if not widely distributed. So the race will be on. And I say will be on because, like I said, sooner or later this will occur. So for us now to have the technologies in place is like getting ready for Katrina. There are antiviral medications that are effective to prevent and treat pandemic influenza. Some of these shown here. Most notably, Tamiflu. You will notice that during the previous 2009 possible pandemic, Tamiflu nearly sold off the shelves. So usually all four of these that have been approved by the FDA work against influenza A viruses, but drugs might not always work because influenza virus strains can become resistant to one or more of these medications. For example, the H5N1 virus identified in humans in Asia in 2004 and 2005 have been resistant to two of these drugs. So monitoring of avian viruses for resistance must be maintained as well. So what about the swine flu? There was a pandemic, H1N1, in 2009, dubbed the swine flu. How dangerous was it? What is the weather report for an emerging pandemic? We have already seen circulation of West Nile virus, SARS, avian flu, the swine flu, and currently the situation with H5N1. The West Nile virus emerged out of COVID as a crossover virus. And it was first identified actually in Uganda in Eastern Africa in about 1937. It was first discovered in the United States in the summer of 1999 in New York State. Since then, the virus has spread throughout the United States. And West Nile is a type of virus that is known as a flavivirus, and it's believed to be spread when a mosquito bites an infected bird and then bites a human. So mosquito control has been one way of combating the spread of the West Nile virus. And we also saw the emergence of severe acute respiratory disorder, or SARS, as probably the first virus that was known recently in history to jump into humans. So a jumping global pandemic of this century occurred with the emergence of SARS in Asia. And it was noted as the 21st century plague. And the story of SARS and its spread is, I think, a shot over the bow. There were 8,422 cases reported since its emergence in 2003 and 916 deaths. So this disease causes a serious form of pneumonia presentation. The infection with the SARS virus causes this disease, and it's a coronavirus. So the adaptation of the SARS coronavirus to humans as sort of a depiction of what can happen as one of these viruses crosses over from an avian source into humans is sort of a harbinger of what could happen with the influenza A virus. So viral hosts of pandemic is a depiction of evolution of the virus itself and how they move, both through their avian sources or bats or other host organisms that harbor these organisms that will undergo mutation of their genome until eventually they have the ability to infect humans. So West Nile virus, avian flu, SARS are all good examples of how emerging viral pathogens can develop in their animal hosts until an eventual crossover event occurs for making them more viable for human transmission. We also see that with SARS and the H1N1 virus, that first responders are at risk. This was also well depicted in the Contagion movie, where another one of the heroes in that film, a physician who we saw doing this report at the Minnesota Department of Health, eventually herself becomes infected. And you can imagine that the internist who is responding to all of these patients who are coming in with the flu obviously is going to be heavily affected. Not only that, the people who go to hospital with that illness or go to hospital for some other reason, as people do every day. And a hospital is a great place to get sick, as we all know. So if the hospital itself becomes infected, first responders become infected, first responders then co-infect other uninfected patients, and away you go. How will a global pandemic move? Well, you can look at this article from the journal Science in September 2005, looking at how avian viruses move throughout their host in China. And you can see the rapidity of the graph shown superimposed there on the global update on the number of cases that are reported from avian transmission. Once these cases of reports start to rise, so should our alert levels. But how will a global pandemic move? Well, migratory bird flyways. This slide here in 2005 on the H5N1 shown in the upper right panel shows migratory major flyways of birds, and pay attention to the red and the blue lines there that traverse hemispheres. So some of those actually are transcontinental, and you can see then that our migratory birds present a very viable opportunity for H5N1 to spread cross-continentally as well as throughout the world, even from Southeast Asia into North America, as shown by that blue line coming from East Asia and Australian flyways. But really, not only just migratory bird flyways, but perhaps more importantly these days, let's face it, migratory human flyways. You get in that little juice can with wings, and one person is infected and 150 people leave that, and not only are they on another continent, but now they all themselves have become vectors. This shows the number of flights coming out of Sydney during one day. And as you can see, that would pose a vector that emerging out of that particular airport would have the capacity of infecting a large number of cities in Australia alone. But let's look at the global connectivity of migratory human flyways. This shows 58,000 flight routes that occur daily throughout our planet now. So you can see the hot spots of the United States, particularly on the East Coast, and then in Europe and in Southeast Asia. And let's face it, we're all very connected and we're a global community very quickly. So migratory bird pathways form the basis of a network for hosting the initial phase of a pandemic. And once it emerges as human-to-human contact, the depiction that is shown in Contagion becomes all the more real. This shows that travel time to major cities in hours and days along the migratory silverbird flyway. And you can see that the hotspots depicted there by the lighter colors showing up corresponding to how quickly a virus might spread in days. So that in less than 8 days, there is a confluence of possible infectivity throughout the United States, Lower Canada, Mexico, all parts of Europe, India, Southeast Asia, and Brazil and parts of South America. All right, we know that now, that there are viruses now that are in process of emergence. We know that their pathway for conductivity throughout the globe is imminent. What can we do? Well, this article put out by a retired American living in the French countryside, David Fenson, describes what we might do for pandemic preparedness. Let's again put this into historical context. The Spanish flu of 1918 killed 50, maybe 100 million people worldwide. We never really will know that number. Maybe 675,000 died in the United States alone. And like I said, this virus not only emerged very quickly, but its presentation was also deadly very quickly. You were sick one day and pretty much dead the next day. What made the 1918 virus so deadly? Well, its virulence has been described as being 50 times higher than, say, a contemporary strain that has been called the Texas virus. 13% of body weight is lost by mice 2 days after infection with the 1918 virus. Weight loss is only transient in mice infected with a seasonal influenza. 39,000 times more virus particles are found in mouse lungs 4 days after infection with the 1918 flu. All mice died within 6 days of infection with the 1918 flu, and none died from the Texas strain. Why should we care about the 1918 flu? Well, it's past, right? It burned itself out. Well, the 1918 flu virus was resurrected. Recently, in experiments conducted on material recovered from a flu victim that was frozen in Alaska permafrost since she died there in 1918, served as a source for fragments of RNA that were retrieved from samples of lung tissue and converted into DNA using RT-PCR, and overlapping sequences then were pieced together to give the full genome sequence. So a DNA version then was synthesized in the laboratory. That DNA was injected into human kidney cells, which produced tens of thousands of virus particles that were used to infect mice. The virus was isolated from those cells to inject into the mice, and all of the mice that were infected with the resurrected virus died within 6 days. So you might say, did we really need to do this? Why not leave a dead virus alone? Or perhaps send it to the sun? Could it be used as a recipe for a future disaster? In the wrong hands, this data is now published. You could go to the data, synthesize your own virus, test it on its infectivity amongst mice, couldn't you? Could it be a recipe then for bioterrorists? I will get into this later when we talk a little bit about dual-use knowledge. What would happen if this virus then escaped from well-intentioned laboratories to reinfect the world again? This is not the stuff of science fiction movies. This actually is printed data. So in the issue of the debate over this publication that occurred in Nature in October of 2005, this quote, "They have constructed a virus that is perhaps the most effective bioweapon known." But the other side of this coin, what can we learn from this? What can we learn about what made the 1918 virus so transmissible and so deadly? Could this help us prevent a future disaster? Well, in the belly of the beast, the secrets of the 1918 flu have been revealed, and the characterization of that virus focuses not only on its transmissibility and its infectivity of human lung tissue, but also characteristics of the polymerase genes that were part of the virulence connected with that virus. But this does bring up the responsibility of scientists and of science. Like I said, should we do this research? Should we publish such research? You could just stick your head in the sand and wait for the next virus to come along and then scramble like hell to try to get things under control. Or you could learn from this and try to develop appropriate strategies that would combat the next pandemic when it occurs, not if it occurs. Could the world stop it even if it could? If you now have this knowledge, could we prevent a scenario that's depicted in the film? Some people say better never than late. Other people would argue that biologists have been asked to breed a culture of responsibility in the face of bioterrorism. It has been said that maybe synthetic biology may be a bigger threat to the world than nuclear war. Many biologists still do not believe that their work could possibly be misused. How could that possibly be? As I said, you could take this sequence and synthesize it yourself if you were so trained, and you wouldn't even need very sophisticated equipment. Certainly you'd need sophisticated equipment, but not nearly that type or to the level that's required to make a nuclear warhead. And when it happens, will we actually be able to contain it? We have more tools in the box than we had in 1918. That was an unmitigated disaster. But will the drugs that we have work? Will the virus mutate faster than we can develop drugs or vaccines? Will there be enough? If people hoard Tamiflu or other drugs, certain countries will have it while the rest of the wildfire rages on. Who will get them? The privileged? The people with money? Certainly the countries with access? This was asked in this paper by Neil Ferguson. When it happens, will we be able to contain it? This was started with a series of papers published in Nature in 2005 about strategies for containing an emerging influenza pandemic as it may play out in Southeast Asia. And so this bottom panel here shows an avian flu epidemic as it would occur in Thailand, unmitigated. And the purple spots, you can barely make it out day 60, but by day 140 have virtually occluded the landscape. And he went on to model the population in Thailand, looking at centers of high population and age distribution that becomes important during the spread of pandemic. So graph B there is the distribution of age groups in Thailand. And the household size is shown in panel C below that. And this also would play out in the transmissibility of a virus as it might emerge in Thailand unmitigated. In panel A here, we see a time sequence of an epidemic showing spread in a single simulation with an epidemic of an R0 of about 1.5. And the red dots show emergence of the pandemic and the days from 30 to 120 when it is raging in the most densely populated regions of the country. Green that follows depicts people who have either recovered from the infection or are dead. So preparing for the next pandemic not only requires an adequate supply of drugs, but an ability to rapidly make vaccines or antibodies that could be used for passive immunity. So most virologists and people in infectious disease believe that it's actually only a matter of time until the next lethal influenza pandemic occurs. The severity of the next pandemic can't be predicted, but from these modeling studies such as Neil Ferguson have conducted, that suggest that the impact of a pandemic on the United States and globally would be substantial, to say the least. In the absence of any control measures, vaccines or drugs, it's been estimated that the United States, under a medium-level pandemic, could cause 89,000 to 207 deaths. Realize that the entire Vietnam conflict caused about 67,000 US military deaths. 314,000 and 734,000 hospitalizations, 18 to 42 million outpatient visits, and another 20 to 47 million people being sick. So that number of people being sick alone has a serious impact on the economy over a period of time that could span from October until April. So the economic impact could range from 71.3 to $166.5 billion US. Influenza pandemics are different from many of the threats for which public health and healthcare systems are currently planning. So to recognize what needs to be done for pandemic preparedness is a situation that the public should be aware of now and actively supporting rather than scrambling at the end and waiting and asking why these scientists didn't do something before this happened. Anybody can look back and say that. And anybody can look at the swine flu and said, gee, they made a lot about it, you see, and nothing really happened. People sequestered things in their homes, people bought guns, people looked at a doomsday scenario. But really, from a scientific standpoint, we should be supporting research on vaccine development, new drug development, and preparedness. I'll get back to this issue when we talk in the next part of the lecture about bioweapons. Because of these differences in the expected size and impact of the influenza pandemic, it's important to plan preparedness activities now that may permit a prompt and effective public health response. So it's up to the U.S. Department of Health and Human Services to support areas of surveillance and detection as well as vaccine development and production. But shouldn't we also have a component of the private sector involved that will economically benefit from this? Certainly the drug makers and the vaccine developers should have a stake involved if a pandemic should occur. That would be my own point of view, I should say. So is this science fiction? This couldn't really happen, right? How accurate is it? Let's just go back to the movie for just a second here. And we zoom ahead towards the end of the film. Again, I don't want to be a spoiler, but this really doesn't ruin anything for you. They're talking about vaccine development at this point, and some of the depictions of empty places around the world. You can imagine how this scenario plays out. The fear, the looting, people entering households, people sequestering themselves away as they are themselves uninfected yet. People who would loot, people who would conduct dangerous activities. Eventually, the fire would burn out. But here, this is actually the end of the movie. We show what happened before the woman coughed. There's a guy working in a kitchen. He shakes hands with the woman that was coughing. Day one, taking the picture. Before that, let me get the right place here. There's a bulldozer with the company's name on it shown here. And it appears to be in a banana plantation or in a forest somewhere in Southeast Asia. There are palm trees. The bulldozer runs into a palm tree and bats fly out. The bats are eating bananas. There it is, it's a fruit bat of some sort. Eats a piece of banana, flies away. The bat flies over and into what appears to be a barn, drops it into a place where pigs are eating. The pig eats the banana. The farmer collects the pig, brings it to harvest. The pig then goes to the restaurant in the back of this truck, prepared on the counter by the chef, who then shakes the woman's hand. Day one. So that's a very accurate depiction about how a virus might cross over. So it's important right now in fact, it is only a matter of time. That surveillance of animal influenza is part of pandemic preparedness. In 2009, the H1N1 pandemic was not as severe as initially feared, and this led to complacency in some quarters that future pandemics will have a comparable impact as the swine flu and could be readily dealt with. I don't think that we should be lulled into such complacency. Neither biology nor history serves us well to come up with that level of thought. So a more virulent virus, such as the one comparable to the 1918 H1N1 or the H5N1 bird flu that is now circulating, could be the cause of a global catastrophe. The slide here shows a depiction of the virus, the H5N1, a microscopic view fused to the head of a ferret. The reason being is that ferrets are used as the model for human transmissibility to evaluate the possibility of an emerging pandemic. The bird flu controversy. Does forewarned equal forearmed with lab-made avian influenza strains? Without question, the H5N1 kills people now. Many of those that it sickens have died, but it does not spread readily amongst humans yet. And in the last bird flu controversy, where H5N1 started to circulate amongst birds, the United Kingdom culled many flocks that contained it, suppressing what appeared to be the emergence of that flu virus at that time. So again, the model that is used for evaluating transmissibility is the ferret, shown in this depiction from Science, February 2012. Meanwhile, two virologists, one Ron Fauché, working in Rotterdam in the Netherlands, conducted a study on the mutability of the H5N1 to determine which genes would be involved in mutation that would allow that virus to become more transmissible from avian sources to human and then human to human. What was discovered actually spurred on quite a controversy. Another researcher at the University of Wisconsin came up with similar results, but what they determined was a very low number of mutations was necessary to cause the H5N1 to be now highly transmissible. And this spurred a big controversy in publication, both in Nature and in Science, when they submitted this for publication last year. Should this be allowed? For one, aren't we telling somebody how to make a bioweapon? On the other hand, similar to what I described for the evaluation of the 1918 flu, could this help now allow us to be better prepared? So this brings on this idea of dual use. And I think this is a good point in this course to bring this up, because isn't this true now for many areas of technology? Whether this is nanotechnologies, as I brought up before, about future use of nanotechnologies going forward and about how that possibly could be misused. Genomics is about how that information could be misused by insurance companies, but certainly forward-reaching, how this would affect dual use in many areas of technology. Some people say that this is a two-edged sword. Well, isn't technology in many cases a two-edged sword? Some people would say that we would be better off if we didn't have the internet. You know, I think that's ridiculous, but some people say we should go back to easier times. Well, you could make that argument. But on we go, and technology does have this dual-use component. And a pandemic is, in fact, only a matter of time. You can stick your head in the sand and say ignorance is bliss until that virus washes over you. Biosafety considerations and appropriate biosafety laboratory level 4 situations are necessary in order to safely conduct and contain this type of research. And certainly BSL 4 level laboratories are not common. And biosecurity as well as biosafety concerns have to be strictly maintained and audited during this research so that you, in fact, do not come up with the ability to generate this in your garage over the weekend. So there's an obligation to prevent next dual-use controversies, and this is part of a policy forum which should be part also of the public debate. If we do not understand both the risks and the benefits, how can we actively have an appropriate policy forum on the topic? If you don't think we need this research, I think you're kidding yourself. If you think that this could not possibly be used as a bioweapon, I think you're kidding yourself. So to have this debate and have appropriate measures in place to both study and control this type of research is increasingly more important. To develop appropriate models and to evaluate them, such as the ferret model that is less expensive than a primate model, but effective at doing this type of research, also has to be maintained. There was a moratorium that came about because of the proposed research done by Ron Fauché and others that we should wait and consider whether or not this should be published. Well, the longer you wait, the longer you wait, isn't it? And so recently, as recent as just a few months ago, this moratorium has been lifted. Well, it is only a matter of time, and I would suggest you see this film and evaluate it for yourself. But again, this is a naturally occurring virus in the movie Contagion. Now we enter into the ability that we can really synthesize pieces of DNA de novo. And the polio virus was synthesized several years ago now from scratch and found to work in models. So the ability to synthesize viruses at will and actually to use synthetic biology to make viruses, whether out of piecemeal parts that exist or known knowledge, is certainly within grasp now. Let alone, again, if we go back to putting on our futuristic goggles, saying what will be the possibility future. This novel by Margaret Atwood, Oryx and Crake, takes a futuristic view of a possible pharmaceutical giant gone awry who purposefully makes a virus at the same time it makes the appropriate vaccine, launching the virus just ahead of the vaccine. But I won't be a spoiler for this one either, but suggest that you read it. I love Margaret Atwood and all of her previous works, but I think you would find this one interesting in terms of the context of an emergent pandemic. Some say the world will end in fire. Some say in ice. From what I've tasted of desire, I hold with those who favor fire. But if it had to perish twice, I think I know enough of hate to say that for destruction, ice is also great and would suffice. Thank you.